I'm sorry to say that I'm going to start my presentation with a very sad note. Yesterday we have lost Dr. APJ Abdul Kalam, former President of India. He was known as Missile Man of India and a great eminent scientist. So today I dedicate my presentation to the great soul. Well, I, Saurabh Chaudhary, research scholar from Guru Gobind Singh Prastha University, presenting my work entitled De Novo Transcriptome Assembly and Identification of Cold and Freeze Responsive Genes in C. buckthorn. Well, first of all, I would like to introduce my subject plant, that is C. buckthorn. It's a hardy deciduous shrub, belongs to the family Alignaceae, bearing yellow to orange berries, which have tremendous medicinal nutritional values. Indian Himalayas is the second richest source in the world of C. buckthorn. Beside medicinal and nutritional value, it has ecological importance also, prevents soil erosion and helps in land reclamations. Besides this, it grows in extreme environments, so it may be important source for new genes alleles for abiotic stress tolerance. To keep this in mind, I have been assigned with three objectives. One is C. buckthorn transcriptome assembly and analysis using NGS, deep stage-based identification of differentially expressed gene during cold and freeze stress, validation of selected cold and freeze responsive gene using QRT-PCR. Well, for transcriptome sequencing, development of C. buckthorn seedlings. We developed C. buckthorn seedlings in our laboratory. Seeds were surface sterilized and germinated hydroponically at room temperature. And after 3 days of germination, seedlings were transferred in growth chamber with 28 degrees Celsius relative humidity and 55% and photo period of 60 hours light and 8 hours dark. The second step is total RNA isolation and quality control. Total RNA isolated using modified CTAB method, which was modified in our laboratory, standardized in our laboratory in 2009, because C. buckthorn contains high secondary metabolites and bioactive compounds. Therefore, it was really difficult for us to isolate the RNA, good quality RNA. So we standardized a method which is modified CTAB method. The good quality and high yield of total RNA was obtained from leaf and root tissues, and RIN value more than 8 were further processed for next generation sequencing. For the transcriptome assembly, high throughput next generation sequencing on Illumina Alexa platform was used, and it was outsourced, and we have received raw data, raw short reads, which was filtered out using NGS QC toolkit developed by Jain et al. In 2011. And this is a summary of the results, short reads which we obtained, that is total number of reads from leaf and root tissues, and high quality reads and the percentage of high quality reads. Approximately 94 million short reads were generated from the Illumina Alexa sequencing, and these were submitted to NCBI short read archive. Out of that, approximately 86 million reads of high quality were further taken as for assembly. Now, the biggest challenge against us was how to assemble these huge amount of data, huge amount of short reads. So we used different assemblers, and for the optimization of transcriptome assembly, including Velvet, Oasis, Abyss, SOAP de novo, CLC Genomics Workbench, and Trinity. Well, for short read assemblers that perform assembly as a function of k-mer, lower the k-mer, higher is the computer's RAM requirement. Thus, for RAM intensive assembly tools, high quality reads obtained from Illumina platform were fed to CD hit 454 suite to remove redundancy, and obtained about 24 million non-redundant short reads. So we analyze both non-redundant as well as redundant short reads with this, and in the first approach we compare de novo assembly as a function of best k-mer. In that case, There are total number of contigs shown by these bold bars, and average read length and N50 read lengths. On the basis of these, we found that OSIS and ABIS be most appropriate tool for assembly in this case. But as we proceed to second approach, that is comparison of assembly as a function of additive K-mer, then we have found the similar results that ABIS and non-redundant ABIS, OSIS and non-redundant OSIS was to be best suited. Then we use another approach in which we compare de novo assembly as a function of additive K-mer followed by TGICL suit. TGICL suit is a is a suit in which long reads were taken for that, so that we use additive K-mers in TGICL suit. In this, it's a two-step strategy, and we assemble the long reads. In this, you can see that again ABIS and non-redundant ABIS, OSIS and non-redundant OSIS is found to be the most suited, with the average read length and N50 read lengths increases significantly in these cases when we use this additive K-mer approach followed by TGICL suit. So after this, we compare the de novo assembly as a function of ability of short read to map back onto the buckthorn transcriptome. In this, we map back all the unigenes received from the different assemblers to the buckthorn transcriptome, and we found 90% hit with the existing transcriptome, except in case of CLC Genomics and Trinity. These are the results of various assembly parameters. In case of ABIS, we obtained 88,297 unigenes, and in case of OSIS, 86,124 unigenes. Well, this also N50 and average read length also increases significantly in these two cases. Now these are the summary of my result. On the basis of various assembly parameters, ABIS was found to be the most promising assembly software, which resulted into 88,297 putative unigenes in buckthorn. BLASTX algorithm was to annotate buckthorn transcriptome. In this, 41340 had significantly the existent non-redundant protein database of NCBI. Higher sequence homology was found with Vitis vinifera, followed by Racinus and Populus. BLAST2GEO suite was used to assign gene ontology terms, and functional annotation revealed that 3,035 unigenes to be involved in different stages in buckthorn transcripts. Many transcription factors and SSR motifs present in the transcriptome also identified. These are the top hit sequence similarity, with Vitis vinifera is the most one. Gene ontology classification, biological process. This is on cellular component basis, and then molecular function. The work is published in PLOS in 2013, optimization of de novo short-read assembly of buckthorn transcriptome. Now, for my second objective, that is identification of cold and freeze responsive genes, we used deep SAGE, a combination of classical SAGE and next generation sequencing used for global gene expression analysis under certain environment. It is also known as digital gene expression profiling. For digital gene expression profiling, again, buckthorn seedlings were grown, and in that case, different cold and freeze temperature were given to the seedlings, that is at 4 degree, and freeze treatment at -10 degree. Now, again, total RNA was isolated, and the 17 base pair tag were generated as per the manufacturer protocol. In case of digital gene expression analysis, tags were directly sequenced using NGS Illumina Alexa platform, and analysis were done, which results into a large number of differentially expressed genes in response to cold and freeze stress. Now, differentially expressed genes were annotated by sequence similarity BLAST with our existing buckthorn transcriptome, and around 450 genes identified responsive to cold and freeze stress in sea buckthorn. This is the pipeline for bioinformatics analysis of raw sequence reads in DeepSage, where raw sequence data were converted into clean text, then gene annotation and normalization, and then screening of differentially expressed gene was performed. This is a distribution of total clean text and distinct clean text received in DGE sequencing. Now, the result of differentially expressed genes, normal versus cold, we have identified 2051 genes upregulated and 1821 downregulated. In case of normal versus freeze test, 2278 were upregulated and 1774 were downregulated. We also compared cold and freeze stress conditions, and we found 2210 upregulated and 1788 downregulated genes. Gene ontology and KEK pathway analysis were also performed for differentially expressed gene. Geoterms suggested metabolic, cellular, primary metabolic, cellular metabolic, and macromolecule metabolic process genes were found to be abundant under cold and freeze stress. 28% and 15% of differentially expressed genes were characterized as gene responsive to stimulus and stress respectively. Photosynthetic, carotenoid biosynthesis, and plant hormone signal transduction, vitamin B6 metabolism pathways was found to be most significantly enriched pathway during cold and freeze stress in sea buckthorn. Now, the result of DeepSage were validated by qRT-PCR. The differentially expressed gene blast with annotated unigenes of sea buckthorn. 22 genes from DeepSage data were selected, involved in cold and freeze stress for validation. The sea buckthorn seedlings were again given the cold and freeze stress for real-time PCR expression. These are the expression of all the 22 genes which used in qRT-PCR expression analysis, MAP2K, ADC, PP2C, AP2ERF transcription factor, RPL50, ATP19, SOR, HSP70, WD40, KC, and PEP. And these are the fold changes. Some are upregulated and few are downregulated. This is the expression profiling of genes under using qRT-PCR. And the work is published in PLOS ONE this year only. So from the study, we have concluded the standardized the high quality RNA isolation from sea buckthorn and established a sea buckthorn seedling in sea laboratory. World Transcriptome data obtained using NGS platform provides an upper age as reference data sets for further studies. Unigenes with no relevant hit in NCBI non-redundant protein database were considered to be sea buckthorn specific and can further be characterized in future. Standardization of assembly tool for NGS data helps further in analysis of NGS data for other species. We also identify 7421 putative sea buckthorn transcription factor genes, developed USMM unigene specific microsatellite markers for sea buckthorn. Many abiotic stress responsivity, especially for cold and freeze tolerant in sea buckthorn, have been identified. Research leads: genes identified to be involved in cold tolerance need to be further characterized. Some of these be explored for development of cold tolerant transgenics. Assembler ABIS optimized for NGS assembly can be used for further similar studies. And in the last, I would like to thank Department of Biotechnology, Government of India to provide the fund for this project, ITS, DST, Government of India for providing me travel grant, and my university, Guru Gobind Singh Pasta University, and of course, Omics International Conference to represent my work here. Thank you.
All right, do we have any questions? Any comments or questions? Okay, good.
It was a nice talk. Could you please tell us about the medicinal importance of this plant?
Yeah, actually, there are a number of therapeutic and medicinal uses of this plant, including it is used as a UV protectant in anti-cancerous. It has anti-cancerous property also, and there are lots of more pharmacological importance has developed. Like the main is anti-cancer. It is contains antioxidants, number of antioxidants it contains, and every part of sea buckthorn, like leaves, fruit, stems, and roots, every part of sea buckthorn is used in medicinal and nutrition.
Is it available in the market?
Yes, it is available in the market. Where the DRDO lab India has first discovered is juice, sea buckthorn juice, known as Leh berry juice. DRDO lab situated in Leh, Ladakh, and my colleagues from China and Russia, they must knowing this plant very well, because it grows abundantly in Russia, China, and of course in the Indian Himalayas region.
Thank you.
Yeah. Okay. Thank you.